Hello all. Today's lesson is on types of selection, which is chapter 9.4 in the textbook. This chapter is about directional and stabilising selection. We will also be looking at required practical number 6. Even though you are not doing practicals at school, we still need to be going through them because they do make up a big part of paper 3. We will do the prac when we get back to school, don't worry, but this is to introduce you to the ideas and the techniques. One of the key things you need to take away from this lesson today is that directional and stabilising selection are two different types of natural selection. So there is natural selection, which is what we described last week. And natural selection can take the form of directional or stabilising selection. It can also take the form of disruptive selection, which I'll also talk about, but the key ones are directional and stabilising selection. So to start us off, if we could please all have a pen and a piece of paper ready, and can you please tackle this exam question? It's a total of 4 marks, so if you give yourself 5 minutes to do this question, that should just about do it. So if you pause the screen now, please do the starter question. Okay, let's have a look at the answers. So what is meant by genetic diversity? The mark scheme says difference in DNA or base sequence, differences in alleles, genes, or gene pool. What I would like you to be saying is genetic diversity is the difference in alleles in a population. So please include that word population. Difference in alleles in a population. Apart from genetic factors, you're looking at environmental factors. So that's really tapping into your GCSE knowledge. What are the factors that affect things? Well, it's going to be your genetic and environmental. So environmental factors are going to be doing that. And the question was, the spotted owl is a bird. They have decreased. What's happening to the genetic diversity? Well, the genetic diversity is going to be reduced. And it says, explain how this decrease may affect genetic diversity. So you say it's reduced. Explain that because there are fewer different types of alleles, or there is a reduced gene pool. It means the same thing. All right, so when talking about directional selection today, we are going to be looking specifically at the example of bacteria in natural selection. So before we start, I would like you to watch this video over here. It's a TED Ed video, and it will hopefully remind you of how natural selection works in bacterial populations to make bacteria antibiotic resistant. So if you could watch that video now, you need to open up the actual PowerPoint to click on the link. So go ahead with that. Okay, so hopefully this is what you remembered from GCSE and what the video has reminded you of. If you have a population of bacteria growing in your body and they are making you sick and you take an antibiotic, those antibiotics are going to kill those bacteria. The way antibiotics usually work is by in some way interfering with the way that the bacteria make the bacterial cell wall. There are different kinds of antibiotics and they have different mechanisms of action. But if you could imagine that this antibiotic here is penicillin, and this penicillin stops these bacteria from being able to make their cell walls. That means when they reproduce by binary fission, they can't make cell walls, so they cannot actually reproduce completely. They cannot make successful bacteria. Within this population of bacteria, there may be 2 or 3 bacteria that have got a mutation. That mutation may confer an advantage to those bacteria. So it may be that the mutation in these bacteria enables them to break down penicillin. So it actually breaks it down. So the penicillin is not effective. So if you imagine you take your penicillin and the penicillin kills all of the yellow bacteria that do not have the mutation, but they leave behind the bacteria that do have the mutation. So they survive, the red ones survive. So if you imagine, this is what you're left with at the end. When those populations grow up again, so you stop taking your antibiotics, they grow up again, so they all replicate. And then you take another round of antibiotics. When you take another round of antibiotics, what you're going to be left with is this. Okay, and then possibly you get sick again because these are still left over in your body and they grow up and they multiply. Take another round of antibiotics and you end up with this. And so the population of bacteria now primarily have got this mutant allele. So we've had a complete change in allele frequency from this population, to this population. Okay, so you've got a population of susceptible bacteria turning into a population of resistant bacteria. So you've had a change in that allele frequency. Okay, this is called directional selection. Why is it called a directional selection? So here are some graphs to show directional selection, but I'm actually going to go to our textbook to have a look at that because they've got quite a nice graph that we can use. So if we have a look over here, so this is your textbook, page 232. So when we talk about selection, and you've seen this before, you've seen us use a normal distribution curve. If we look at the axes, on the axes over here, we've got whether or not the bacteria are resistant to antibiotics. So this could also be represented by called allele frequency of the antibiotic resistant gene. Okay, so low antibiotic resistance, high antibiotic resistance. And here you have the number of individuals. So if we think back to our slide, the original population might have had a distribution like this. Very few bacteria with a lower antibiotic resistance, and then you've got the mean amount, and then you've got very high antibiotic resistance at this end. So that when you take the penicillin, all of these bacteria would be killed, but only these ones over here, maybe they survive because they are the most resistant. And then the new population will grow up from these bacteria, from this limited population over here. And when that population grows up, it has a completely differently placed normal distribution curve that has shifted along to having high antibiotic resistance. Okay, so the use of the antibiotics puts directional selection pressure on the bacterial population. The key thing around directional selection is that the extremities of the curve are selected for. So the extreme end, higher resistance of the original curve is selected for. So it pushes the population in a new direction, the direction of high antibiotic resistance. Okay. So let's just go back to my other page. Where are we? Here we go. So hang on, bring my shared window to the front. Sorry, I'm just figuring out my sharing. There we go. Okay. So the selection pressure is essentially pushing the curve in that direction. It's pushing, so here's for bacteria specifically, pushing the bacteria in the direction of being highly drug resistant, from low drug resistance to high drug resistant. Okay, so that's directional selection. And there are lots of different examples of directional selection. And I'm sure you'll be able to think about that as you work through the chapter. Couple more useful links. These are specifically about antibiotic resistance if you're interested in finding out a little bit more. Stabilising selection is a different type of selection. Stabilising selection, in fact, selects for the mean. So directional selection pushes the curve in one particular direction and it selects for extremes. Directional selection happens when the environment is changing. Stabilizing selection, however, selects for the mean, so it selects for the mean. And stabilizing selection happens when the environment is not changing. So when the environment is stable. So if the classic example is birth weight of a baby. So if we look at the birth weight of a baby, some babies can be born at 2 pounds, extremely tiny, okay? But on average, where's my little spotlight gone? On average, but on average, the mean mass of a baby, mean birth weight, is going to be about 7, 8 pounds. Maybe if you speak to your parents, many of you would have been born around that weight. And then, of course, there are bigger babies, right? Now, why does the curve look like this? Well, if we look at another graph that's imposed on here, and we look at infant mortality, we can see that for infants that are born at a very, very low birth weight, they have a very slim chance of survival. And that chance of survival will increase as the baby gains more and more mass. So by the time you get to about a 6 pound baby, then you're pretty safe that the baby is going to survive. But then as the baby gets bigger and bigger, again, the percentage of mortality starts to increase. And this is going to be about the fact that there are going to be birth complications if the baby is too big. And so there are lots of reasons why women would have died of childbirth before medical intervention. And one of them is that the baby is too big, essentially, for the mother to give birth without a cesarean section. So that would end up with mother and child dying. So stabilizing selection, classic example, birth weight in humans, the environment is not changing and the selection is for the mean value. Okay, directional selection, environment is changing, selection is for extreme ends. Those are the two main types. The third type is called disruptive selection. It's not part of our curriculum, but it's quite interesting. I think we might have mentioned this before. So if we have a look at this graph over here, if this is our mean curve, our normal distribution curve, there's our normal distribution curve, there would have been the mean value of that normal distribution curve. So imagine now, this is about size of male salmon, size of male coho salmon, right? So there is the mean size of the male salmon. That's a large salmon. That's a small salmon. Okay. If we're looking at the males that are most successful when it comes to being able to fertilize eggs, we find that the larger males are very successful because they're large and they're strong and they can impress females by their strength. But we also find that the smaller ones are successful because they can sneak in and they can fertilize the female's eggs while no one is looking. So you end up with the mean being selected against. So the selection is for the little sneaky fish, and then the middle-sized male doesn't really get selected for because he doesn't stand much of a chance, and then you select for the larger male for his largeness and his prowess, basically his strength. So that's disruptive selection, and here's just a few more examples so you can have a look at those, give you an idea of directional, stabilizing, and disruptive selection using the idea of tails on animals. So read through that. Okay, and here's a video. So if you could watch this video as well. This is just about 2.5 minutes long, and it will just review the ideas of directional, stabilizing, and disruptive. Don't worry about Hardy-Weinberg. That's for next year. So we will talk about it, but we talk about it next year. They say it a couple of times. It's for next year. Okay, so that's directional and stabilizing selection. Next thing you're going to do, please, is, oh no, one more thing. So the adaptations that any, particularly animals, are going to have can be either anatomical, physiological, or behavioral. Okay, so these adaptations that come through the selections. So anatomical, we're specifically talking about anatomy. And so I've taken examples from your homeworks from last week. A fair few people spoke about giraffes. So the length of a giraffe neck will be a directional selection, selecting for longer neck, and that's an anatomical adaptation, as is the fur length of the, oh, I can't remember, was it called the snub-nosed golden monkey? He's adapted, there is a fur length adaptation here for cold. Physiological adaptations, I've got an echidna here. Echidna is a little marsupial that lives in Australia, New Zealand. I can't remember which one, Australia, I think. And they have fantastic physiological adaptations in that they can burrow underground and they can survive with very low carbon dioxide levels underground for a long time. And they think that it might be part of what makes them successful in surviving bushfires. So their physiology is adapted. And in this example, they can live with very low carbon dioxide levels when they hibernate. And then the adaptations might also be behavioral. So people, there was maybe 1 or 2 essays on wolves, and people spoke about the social nature of wolves. So wolves have adapted to be social animals because of the greater success that you get in hunting in a pack, for example, and being able to claim territories in large packs. So those are the different types of adaptations that you might see. Okay, all right. So that's your that's the selection mostly taken care of. I'm not going to spend a lot of time on the required practical because I'm going to let you watch this video. So this video will take you through exactly what you would have done in the lab. We would have done this exact investigation exactly as it's shown. And so watch that, approximately 8.5 minutes long. And with that practical, there comes a caboodle worksheet. So you will find that on the team, or I'll attach it. And some exam questions. I think there are 3 exam questions that I would like you to answer on these, on required practical 6. Keeping in mind that required practicals are tested in paper 3. So these will all be paper 3 style questions. What I want you to do when looking at this practical is not just think about the actual practical itself and what it's trying to show. So it's working out which is the most powerful antibiotic, but really focus on the methods. So the methods used in this are around sterility. So why using a Bunsen burner flame? How do you sterilize things? Just thinking about the agar plates, why they kept a certain way up. So there's lots of technique in there. Look at those techniques and make a mind map of all the different techniques and why they are important. So in summary, what I'd like you to do, make notes of or a mind map of chapter 9.4. So that's just a case of your normal summarizing notes. On page 234, there is something called Must Be a Cuckoo. Answer that and mark that. You're going to watch the required practical 6 video. Like I said, there's going to be a worksheet for that. So make a mind map, and then there's questions there. Answer questions 1 to 4 on there. The others you can't because you haven't done the practical yourself. Then there are exam questions on required practical 6. Then I want you to do a bit of revision, and to do that, do the end of chapter questions at the end of chapter 9. I'm going to put the answer sheet in Teams, so you're going to use that for revision. And then, because it's the end of the topic and we can't do an end of topic test in the same way, I would like to use Seneca and Caboodle in order to test your knowledge. So I'll be assessing your knowledge. I've given you 2 Seneca tests, and I have given you 2 Caboodle tests. For both of these, you have to log into Seneca, and here you have to log into Caboodle. So I'm just going to see if I can get to Caboodle to show you what it might look like. Let me just have a look. If I can, here we go. Yes, okay. So this is what the test looks like. Genetic diversity and adaptation objective test, And then the other one is genetic diversity and adaptation on your marks, part 3. So I've set these for you, and then you will need to complete that for me so I can see how you are doing. All right, as usual, any questions, please let me know. If you do have questions, I would be really grateful if you could ask them in the post section of the team, because that way everybody can benefit from the answer. There is no such thing as a stupid question. Okay, all right. So good luck, everyone, and I will be in touch. Okay, thank you.